All right, then, welcome to the January meeting of the North Coast chapter of the California Native Plant Society. Glad to see you all out on this dark and stormy night. So I'll start out as Carol usually does and say who has seen some interesting native plants recently that they'd like to share with the group. Anyone? I know it's early in the season.
And very cold outside.
So we're going to pass around. Could you do that, Sean? Just to get that started. This is the list for anyone that wants to join our mailing list to be informed about our various meetings and programs and plant sales and field trips. So if you're not already on our mailing list and you would like to be on the mailing list, you can sign up on this sheet that's going around. Also, if you're interested in volunteering for any of our activities, especially our native plant nursery, then please sign up on that sheet that's going around, and someone will be in touch with you. And we really appreciate all of the volunteers. This is a totally all volunteer organization, and it takes a lot of people. We are a very active chapter. It takes a lot of people to get done all the things that we do in the chapter. So thank you everyone that does volunteer. Does anyone have any announcements? Okay. Wow. Okay.
Please,
Can you stand up, Joanne?
yes. Please eat all the cookies so you don't have to bring them.
Okay. Sorry, people in Zoom land, you can't get you won't get a chance to try some of Joanne's yummy cookies here. Thank you, Frank. Okay, then I'm going to launch into introducing our speaker tonight, Sean Stewart. Sean is a recent graduate from Cal Poly Humboldt with a master's degree in natural resources, environmental science, and management. Before going back to graduate school, Sean worked for about 20 years in the architecture and engineering field as a drafter, designer, and project manager. But he became increasingly concerned about the environmental destruction that he was witnessing with the projects that he was working on. And at one point he decided that he needed to go back to school and study environmental restoration, which he did. And today he is doing several different things. He teaches drafting at College of the Redwoods. And he also has started his a landscape design business, Stewart Native Plant Designs. He's volunteered for some time with our chapter and often goes out on some of the native plant consultations. So please join me in welcoming Sean Stewart.
Thanks, Donna. I actually have a microphone here. So my thesis was pretty darn math heavy. So I hope everybody brought their calculators tonight. Just kidding. I really try to simplify things down so that we could actually enjoy the presentation. So my thesis, really, I did a demographic study of the rare and endangered plant species Astragalus applegate, or Applegate's milkvetch. And I did this in order to come up with some new conservation strategies and also to assess some existing one. So tonight we'll kind of learn more about this special little species here. Now, before I get into the study itself, I need to talk about some background information. We are currently living in a time period known as the Anthropocene, and as its namesake, humans have impacted much of the environmental systems around the globe. oops, that way. Now, as part of these impacts, extinction rates are now 100 to 1,000 times higher than naturally occurring ones. And while that's not really as immediate as a massive meteor hitting the planet, we're still losing species at an astounding rate. This is largely due to anthropogenically caused habitat loss. Here you can see a lot of land use change in the upper Klamath Basin, where Astragalus applegate grows. And you can see a lot of urban areas up here on the top right, and then a lot of conversion into agriculture fields in this kind of mosaic of green here. Also note that the Klamath River down here is largely contained within the agriculture and urban settings. Now this photo is very specific to my study, but we are seeing these type of changes around the world. Now, another thing that is impacting habitats is, that is degrading habitats, is fragmentation. And so this is not the upper Klamath Basin, but it's a good example. And you can see in this middle forest here, the natural resources in it are much more limited than those in the background, more continuous forest. Additionally, any populations that live in this forest here might have a difficult time interacting with any populations living in, let's say, this forest to the right. This causes inbreeding within the populations and reduces fitness levels in the individuals. Now, another way that the habitats are being degraded is by disrupting the natural ecological processes that usually occur. This is usually because of human mismanagement. One example of this is the clams along the dams along the Klamath River. And what this does is it limits the water flow downstream. And this kind of ruins the flooding regimes that happen and that the native plants really have evolved to take advantage of. Now, I'm not saying all this to be a downer or anything like that, but I do feel like if we are a major part of the problem, we also must be part of the solution. And so this is just myself on the right and 3 other like-minded students that helped me out with the projects. Now, rare species are often prime candidates for conservation, and this is because they are often more susceptible to anthropogenic change, often due because of their small population sizes, limited geographic range, and specific habitat requirements. Now, a lot of populations live in habitats with different environmental conditions. And this is especially important because these environments have varying types and varying levels of degradation. So coming up with unique conservation strategies for each population is really critical for the species in general. But before we could really do any conservation, we first must monitor the species and collect long-term demographic data. And what we can do with this demographic data is we can uncover the species needs and the fluctuations within its life cycle. With this demographic data, we build population models, and population models really quantify the vital rates within the life cycle of the species. And vital rates are typically survival, growth, and fecundity, with fecundity just being a more scientific term for reproduction. Now, there are multiple tools that use population models to develop conservation strategies, one of which is we can extract population growth rates from them. We can predict extinction by forecasting these populations into the future. And then we can assess the vital rates influence on population growth rates using an elasticity analysis. Finally, we can assess environmental conditions that affect those vital rates by using a life table response experiment. Now, we'll go into at least a little bit more depth on these analyses a little bit later. As I said before, I also assess an existing strategy being used for Astragalus applegate. And this happens to be a fisher being translocated from captivity into the wild. And translocation, well, many of you have done it when you buy native plants from the CMPS plant sale and put them in your garden, right? So you translocated from a greenhouse into a garden. And translocation has been questioned about its efficacy of protecting these species. This is because rare plant species also often have low survival and fecundity rates. However, reinforcing extant populations seems to be a more promising method of translocation. And just for your own knowledge, extant equals surviving. And so an extant population means it's a surviving population. So why is reinforcement promising? Well, the presence of extant individuals means that the ecological requirements of the species is already on site. Reinforcement also increases plant density, which means that pollinators don't have as far to fly in order to pollinate the next plant. And reinforcement can reduce inbreeding in populations. This is because if you take an individual from a different population and bring it into a new population, you then introduce genetic material into it. Now, the problem with this is sometimes you might lose home field advantage. So we need to take care not to do that. What home field advantage is basically a population has very specific genetic traits that allow them to persist in their own unique microenvironments. Okay, so for those of you that do buy native plants, just try to make sure that they are locally sourced and they're not from like Southern California or anything like that. Now, my thesis had 2 different chapters. And the first chapter, I reviewed some extant populations, and I had really 4 questions. In essence, I wanted to know what the population growth rates were for each population. I wanted to know what the extinction risks were of each population. I wanted to know which vital rates had the most influence on those population growth rates. And I wanted to assess the environmental impacts on the vital rates within each population as well. For my chapter 2, I really wanted to assess reinforcement as a potential conservation strategy for this species. And I did so by figuring out how age influenced 3 different variables and then compared planted and extant individuals to each other. Yeah, so reinforcement, basically we're taking plants that were grown in a greenhouse or another setting, and we are reinforcing an already established population. This really boosts up the number of the population. All right, so let's talk about the Astragalus genus for a little bit. So Astragalus is in the legume or pea family, also known as Fabaceae, and they can be either annual or perennial. And in California alone, there are about 100 species or more than 100 species of Astragalus. And the calscape.org shows actually 177 varieties of Astragalus. So another plug there for California Native Plant Society. And all species in here, the leaves are pinnately compound and they form raceme inflorescences. And so the leaf on the left shows all the little leaflets on it. And then on the right is a newly formed raceme. The flowers of Astragalus are really made of 4 different parts, or at least the showy parts of it. We have the banner or the standard, that's this big part in the background. And then we have the keel in the very middle here, and I kind of think of this as a landing pad for pollinators. And then we have one wing on either side of the keel. And then all the reproductive bits are inside, and I'm not really going to go over those. Now, there's a fun fact. First, the seeds develop inside pods. So all your peas, all your beans, all that good stuff. But there is a fun fact for Astragalus. It is the Greek word for ankle bone. And ankle bones were used to make dice back then. And the genus Astragalus was named so because the seeds inside the pod rattle like dice. So Astragalus has over 3,000 species worldwide. And because it's such a large genus, there are many of those that are endangered. 3 of the ones that I'm going to show right here are native to California. Pearson's Milkvetch, which is only found in the sand dunes of Southern California and Mexico. Coastal Dunes Milkvetch, which it's not Humboldt native, but there are other coastal dunes around California that it resides in. And then the Big Bear Milkvetch, which is only found in the San Bernardino Mountains in Southern California as well. And I just really like this species because those pods really look like they're almost Christmas ornaments, right? Pretty awesome. So let's discuss now Astragalus applegatei. So it historically persisted in those seasonal floodplains of the Upper Klamath Basin. And dams and canals were built in order to control water flow, which reduced flooding throughout much of its historic territory. It is narrowly endemic to that Upper Klamath Basin, meaning it only grows there. And it was thought to be extinct until it was rediscovered in the 1980s. Klamath Falls is about 5 hours away from Arcata as well. There are only 7 known populations left. However, Washburn Railroad here was recently bulldozed to make room for some storage warehouses because people's stuff is more important, right? So this just really highlights the importance of conserving the species on protected land. Now, for those of you that don't know, Oregon does not have laws protecting endangered species on private land. So that's why this was allowed. Astragalus applegate is a perennial herb that loses all of its above ground biomass in the fall, leaving behind a root crown and this deep taproot. And this was kind of a smaller individual, so I can only imagine what the taproot looks like in some of the much bigger individuals. Established individuals then resprout in the spring, although multi-year dormancy can occur. And then seedlings also germinate in the spring, either from seeds produced the previous year or from a persistent seed bank. Here's a great photo of one of the seedlings. The bottom one, not so much, but it does show they're about 3 centimeters high when we find them. They can be pretty hard to spot sometimes. Now, after the established individuals resprout, the stems often kind of spread out along the ground. And usually the newer growth, the newer stems are green, whereas a lot of the older growth at the base of the roots are red. They're quite gorgeous to look at. Now, multiple individuals usually grow in clumps, and this is because there's really no known seed dispersal method. I do kind of imagine that historically, when the floods came, it kind of moved the seeds along, but those floods don't occur in any of the environments anymore, so they grow in clumps now. And like the rest of Astragalus, here are the fruit pods, and the color of these really range from green to this really cool green reddish variation. As the pods mature, they split open, as you can see in kind of these areas here, and then the seeds fall out. Now, I collected these fruits because I was trying to estimate the number of seeds per pod. And here's a closer picture of the seeds themselves. Now, interestingly, all the black seeds are the only ones that are actually viable, that can actually grow seedlings from them. One last thing about Astragalus applegate is that seedling survival really depends on a specific mycorrhizal fungi, and it also produces a nitrogen fixing bacteria as well. And here's the mycorrhiza inside one of the root cells. So I collected data from 4 of the populations: Airport, Collins, Uwana, and Miller. And let's go ahead and go into detail about those. So Airport is the 2nd largest population, and there are many different subpopulations throughout this site. I collected data from the little subpopulation that's right here, but again, it's kind of spread all throughout here. The site is artificially drained, and which is really meant to carry water away as soon as possible for the safety of airport operations. And the whole entire site is also managed by mowing regimes, which is actually beneficial to the species itself as well as the safety of the airport. Here is a ground photo of that subpopulation that I sampled from, and just note a lot of the bare ground and lack of vegetation here. And here is a photo of the mowing regime, which there is actually some problems with this, which I will go into later. Here's one of the other subpopulations. So I was part of a Fish and Wildlife Service project trying to figure out how many individuals are actually in these other subpopulations. So we flagged every single one and then collected flags and counted them. Because it is right next to the airstrip, we had to have a security escort. Now let's talk about Collins. It is the largest population, and there are many subpopulations that are only found in grazing exclusion zones. So in this photo, you can see this kind of line around here, and that's a fence to exclude livestock from this area here where the subpopulation is. And you'll also notice that the Klamath River runs right along here. So the subpopulations like to be next to the water, but I have found some that are about 1,000 meters away. Collins is on private land, meaning that it is not protected by the Endangered Species Act. However, the landowners do have some sort of agreement with Fish and Wildlife Service to protect the subpopulations from livestock. I don't really know any other details besides that, though. Here is a photo from that same subpopulation, and you can see the Klamath River right in the background. And here's another photo of it, and it's really hard to see, but that's Shasta Mountain right there. And on clear days, you always knew which direction south was because you could see Shasta. And here's one of those big clumps of individuals that I was talking about. And this is really nice to see because, well, the plant's endangered and we want to see these big clumps, right? And then, of course, you should always take time to enjoy the wildlife. And this is a photo of a turtle just resting on a log. And that was pretty awesome to see. So now moving on to Uwana, it is one of the smaller populations. It does not have subpopulations. This site is the Uwana Flat Preserve, which is owned by the Nature Conservancy. You can see a big salt flat down here in the south, and the entire site slowly drains towards these water canals meant to move water off site away from this big urban area to the northeast of it. Also note that this is the site of the reinforcement project, which I will cover later on. Now here is a photo of the ground looking south towards the salt flat. And here's an individual that is actually growing in the salt flat. And please note that this is just some really bright red stems all throughout it, and the really pretty purple flowers as well. Here's another photo looking north towards kind of the urban areas, and looks like we're about ready to start measuring some of the plants. And here's an action shot of myself writing down data, and we spent a lot of long hours on our hands and knees for weeks at a time, but it was all worth it. Finally, we have Miller, and this is by far the smallest population, and it really exists in a seasonal wetland in the Miller Island wildlife area. Because it is a seasonal wetland, it is dominated by perennial grasses, which means there's a lot of competition with other species. And I also want to note that there was a failed reinforcement project a couple decades ago here, but it was likely due to drought years and herbivory. And here's a photo of one of the plots here. This was taken before I started the project. So when I finally got out there, these grasses were really tall and much denser as well. Now, in order to collect data at each of these sites, we built 3 to 4 plots in 2016, and then we numbered all individuals inside of them using these metal tags. And this includes all seedlings in subsequent years. Unfortunately, throughout the years, we did lose some of the tags. Some voles often brought the tags underground, which means we couldn't find them. And I do suspect that some birds saw the shining metal tags and probably swooped down and grabbed them, but we still got a lot of data out of this. I did collect demographic data from 2016 to 2020 in June and July. And I surveyed for survival, reproductive status, and collected enough data to estimate the number of seeds per plant. I also took 2 horizontal dimensions to estimate the elliptical canopy area of each individual. Now, with those demographic data, I built population models called integral projection models out of them, or IPMs. Now, we don't need to go into this aspect of it right now. We can talk about that later on if you'd like, and I would love to be honest. But just know that the population models are based on the sizes of the individuals. However, it is important to look at the life cycle of the species within the context of those population models. So in this figure, we have the initial year in the left corner on the left side, and then the following year on the right side. And then here's the symbol for each of the vital rates, okay? And so our first vital rate is the survival and growth of established individuals, or PE. And so what that is if established individuals of a very specific size survived the next year, then it either grew, remained the same size, or shrank. And if those established individuals were reproductive and produced new seeds, then those seeds must survive to the following year and either germinate to create seedlings. I call this fecundity of established individuals, FE. Or the seeds simply survive and go into the seed bank, and I call this growth of the seed bank, GB. Now, the seed bank portion is very similar. If the seeds survive, then they could either germinate, and I call that fecundity of the seed bank, FB, or they just simply survive and remain in the seed bank. I call it survival of the seed bank, SB. So with those models, I just wanted to start answering my questions. So question 1 was, what are the population growth rates of each population? And on this figure, we have y-axis is the population growth, and we use the symbol lambda for that, which that will come into play later on as well. And you can think of lambda as if it equals 1, then the population is stable. If it is greater than 1, then the population is increasing, and less than 1, it is declining. And along the x-axis is the populations, and each symbol references different annual transitions. Now, there are two things I want to point out here. Number one, airports, 3 out of the 4 annual transitions are declining. So that's not good. But second, we have a large spike in the 2018-19 transition at Collins and Uwana. Now, this typically indicates what's called a bonanza year, which is a result of unusually ideal environmental conditions. However, I really couldn't pinpoint why this occurred. And so I simply just don't know. I hypothesize some things, but I don't know. So for question 2, I wanted to know what the extinction risks of each population was. And to do that, I did a stochastic forecasting method. Again, we don't need to go into the math on that, but I did forecast 10 years into the future, and I used a quasi-extinction threshold of 3,000 individuals. And so what this means is if the population ever dips below 3,000 individuals, then Fish and Wildlife Service says that the population is too small to recover from that. So here are the results of this analysis, and on the left is the population. N sub 0 is the initial population size that it started off with, and then lambda sub S is the population growth rate 10 years into the future, and this includes a 95% confidence interval here, and then the extinction risk here all the way to the right. And so we can see the airport barely declined, but had a 0% risk. Collins grew, also not at risk. However, Uwana, it grew, but it was at 36% chance of falling below that 3,000 threshold. Miller was interesting. I didn't have enough annual samples to do this analysis for it, but we already know, we can see it's way below the 3,000 threshold. So the question became, well, why is airport declining but not at risk, whereas Uwana was growing but at risk? And so population size matters. We can see airport was well above that 3,000 threshold, and Uwana was only about 400 individuals to over it. Yeah. Just risk in general. Yeah, so anytime it dipped below that 3,000 threshold during those 10 years, it was deemed quasi-extinct. Yeah. So there's actually some problems with this number. That's going to take some time. But essentially, Fish and Wildlife Service came up with that number because it is the smallest population size in observance before they actually saw the population grow. There are other methods that other people use for quasi-extinction that I won't go into here. Yeah. Back to why Uwana was at risk. So population size matters. And also we see that 2 of the annual transitions were in decline. So it's that low population combined with a 50% chance of decline that made the 36% chance to go quasi-extinct. So what did we learn from those first 2 questions? Well, we know that airports needs to start growing again more often. We know that Uwana, we need to get that population size to increase. And we know that Miller's it really just needs immediate attention because it's not doing very well. So we know which populations need our help. Now the question becomes how. And for a partial answer to that, I move on to question number 3. Which vital rates have the most influence on population growth rates? And I conducted an elasticity analysis to answer this. Again, I can go over more of the math afterwards, but essentially this type of analysis guides conservation towards individuals with very specific characteristics. And to answer, and here's the results of that on the right. but I'm going to take a drink of water. So here's the life cycle and the vital rate. So please memorize that because I will be taking it away.
You're talking about in the wild, or are you talking about a seed bank?
In the wild, yeah. So anything that stays within the soils, yeah. So on the right is the results, like I said, and along the x-axis is each of the vital rates, like right here. And the elasticity value is on the y-axis, and the higher the elasticity value, the more influence it has on population growth. And so it's pretty apparent survival and growth of established individuals had the most influence on population growth rates. Now, what this really means is that large individuals are really important to the species. Not only do larger individuals have higher survival rates, but they produce a lot more seeds as well. So for conservation, we need to make sure that small individuals survive and grow, whereas large individuals, we need to make sure just survive. But how do we do that? So I came up with 3 different conservation strategies based on the natural history of the species. And the first one, every gardener should know about, especially those in this audience, is water addition. So we know that Astragalus applegatei's historical habitat were seasonal floodplains. And I also know that a lot of other species, a lot of other floodplain species have had water addition applied to them, which has increased vital rates. So I suspect we can do the same thing here, especially in drought years. However, we don't really know much outside of that. We really need to know how much water they actually need and if the time of year matters. So is it more important to water them during the spring or during the summer? The next one is mycorrhizal inoculation. Now, from a study from Beretavania et al. In 1998, we know that seedling survival really depends on mycorrhizal fungi. And again, you can see that mycorrhizal fungi in picture 1. And here is the rhizobium bacteria nodule in image 3 that fixates nitrogen to the soil. So other plant species have improved vital rates from mycorrhizal inoculation. I'm hoping that we can do the same thing for Astragalus applegate, assuming we can cultivate the correct mycorrhizae. However, we really don't know a whole lot else about the mycorrhizal fungi besides that. We don't know its impacts on adult individuals, and we don't really know the abundance of the mycorrhizal fungi currently in the soil. So a lot of work needs to be done there. And the final one is protection against herbivory, or just protect the plants from animals, essentially. So throughout the years that I was there and at every single population, I saw leaflet and fruit damage from insects. And here's an example of that. You can see this little hole in the pod, and there were absolutely no seeds left in there. But I also saw a lot of damage from jackrabbits and those type of things. They would eat part of a stem or the whole stem all the way to the root crown. And this is actually a picture of something rather extreme. So all of the stems were eaten all the way down. And unfortunately, this was a rather large individual that I found its root system lying on the ground the following year in my hand right there. So obviously, herbivory can really impact the vital rates of the species. Now, other studies, in fact, a lot of other studies have put cages around individuals. And so I would like to see that happening at some of these populations. So now on to question 4. Which vital rates contributed to the difference in population growth rates between populations. And no, that's a mouthful. There's really no other way to simplify that. But I essentially constructed a life table response experiment for this. And this really compares two populations in order to assess the environmental effects on vital rates. And this type of analysis is really good for population-specific conservation strategies. For this, I'm really going to focus on just Airport and Miller because they were the two most opposite and extreme populations from each other. And here you can see this is Airport versus Miller. And the y-axis is the contribution value. And we have the vital rates here on the right. And we can see that at Airport, survival and growth of established individuals had a negative contribution to the difference in population growth rates. Alternatively, at Airports, we had positive contributions from fecundity compared to Miller. And so what this really means for Airport is that most of the established individuals there were very small, had high mortality rates, but there were a lot of seedlings that were being germinated that made up at least most of the losses of the established individuals. And for Miller, this means that the individuals that were there were very large and they typically survived, but there was virtually no seedlings that happened there that we found. So why is there a difference in survival and growth of established individuals? Well, we can see from this aerial view of Airport, the site is extremely dry compared to the surrounding areas. We also notice that it is very far away from the Klamath River. And then we have Miller that is pretty close to the Klamath River, but also that it resides in the wetlands that is controlled a bit by the Klamath River as well. So what that means is for Airport, in order to increase survival and growth, we need to increase the water retention on site, or as stated before, add some water regimes. Now, why was there a difference in fecundity between the two sites? Well, on the top, again, is that photo of the plot at Miller, and on the bottom is Airport. And at Miller, you can see, again, a lot of those perennial grasses, meaning a lot of competition with other species, and has absolutely no bare ground to it. This combination makes it really difficult for seedlings to establish themselves. And in fact, we only found 6 seedlings at Miller the entire time we were out there. So that's not very good. So how do we reduce that competition? Well, mowing regimes have shown to really mimic the geologic disturbances of flooding. And this works pretty well for Astragalus applegate because this individual here typically grows along the ground, meaning when the mower blades go over the top of it, most of the stems do not get impacted, but all the surrounding vegetation does. Mowing regimes are utilized at Airport and the unstudied OC and E population, but I think it could be done in other places. But this does bring me to a problem with the mowing regimes. So if the surrounding vegetation grows really tall, that means Astragalus applegate also has to grow tall in order to compete for sunlight, like on the left here. And when mowed during these conditions, well, it kind of mimics herbivory. So the frequency and the timing of the mow really matters. The initial mowing should occur before the plants actually resprout in the spring, and then should be a continuous frequency throughout the growing season. So what did we learn from those last 2 questions? Well, survival and growth rates of individuals are extremely important to population growth. This is especially true at Airports. I also hypothesize that water retention, mycorrhizal inoculation, and herbivory protection can increase survival and growth rates. We also know that any population struggling to have seedlings could benefit from mowing regimes to reduce competition. This is especially true at Miller. So now on to my chapter 2. And for this, I wanted to assess whether or not reinforcement was a viable conservation strategy. So 1,322 total individuals were planted between 2012 to 2014 at Uwana Flat Preserve. And here, this series of polygons on the southern portion where they were planted. And then the black squares here are where the extant plots are. Yeah, so when they were grown in the shaded area, they had what's called home soil. So they dug up soil at one of the extant populations and mixed it with potting soil. Yeah, so this is a photo of that. And this is one of the crews that planted everything. Now, I surveyed for, again, survival, reproductive status, and plant size of the planted individuals. And I surveyed them from 2014 to 2023. And then, of course, the extant populations, I did the same, but from 2016 to 2023. Took all that data and I came up with some statistical models that are shown right here. I'm not going to go into the weeds with these models, but I will describe them a little bit. Along the x-axis is the age, and along the y-axis is the demographic variable. The planted group is the red dashed line, and then the extant group is the continuous blue line, and then the shaded areas are the 95% confidence interval for them. Now, for survival percentage, well, simply put, survival was very similar between planted and extant individuals. That's really, really good. For flowering percentage, the only difference was between planted and extant seedlings. You can see planted individuals were typically flowering, whereas extant individuals were not. And this is likely because planted individuals were grown in a much more ideal controlled setting. And that meant that the plants could give more energy into flowering. And yeah, we did find that once individuals were reproductive, they generally stayed reproductive. And now on to the elliptical canopy size. On average, planted individuals were larger, no matter what the age was. However, there really was no statistical difference between the two groups. And just a couple more totals here. By the end of last summer, there were 129 planted individuals that still survived, which is about 10% of the total originally. And then we did find about 122 seedlings that were found like around the perimeter of the planted individuals. This does suggest that there is natural recruitment happening. So what did we learn from that study? Well, we know that all demographic variables were essentially the same, with the sole exception being the flowering percentage of planted and extant seedlings. We know that the planted individuals can persist long-term and that they do have the ability to naturally recruit. And so I think that reinforcement can be a very successful conservation strategy. Now, this also means that there is potential to establish new populations as well, but it really depends on if we can find suitable habitats for that, which I'm not so sure we can. And just for a final word about all of this, I really hope that my thesis assisted in conserving Astragalus applegate. It was a lot of work, and I really hope that these efforts really allow people in the future to enjoy this species. So there was a lot of people that were involved in this study, most notably Carrie Byrne, Dr. Carrie Byrne, She really spearheaded much of this, and I can't thank her enough, but there are a lot of agencies here as well. And then, of course, everybody here. And then finally, I want to thank California Native Plant Society for letting me discuss my thesis here. And thank you all for coming and really helping out with native plants. Thank you very much. All right, any questions?
Hi, Andres here. Do you hear me?
Yeah, we hear you.
Yeah, I have a few questions. One question is, is it this species poisonous for cattle or not?
It is
You
not
check.
poisonous to cattle. In fact, it is a what's considered an ice cream plant. You know, we all like ice cream, and a lot of the mammals out there really like this plant as if it's ice cream.
Yeah, that's the bad fact.
Yeah.
And second, you didn't mention anything about insects pollinating these species. Did you observe any?
I didn't catch that. What was that?
Pollinating insects.
Oh, pollinators. Yeah. So I did show a photo of one of the very important pollinators, which is Melissa blue butterfly. I should have pointed that out. I apologize, but it is a rather important pollinator for the species and is quite beautiful. But also, Astragalus applegate is a very important food source for the larval stage of the Melissa blue butterfly. So there's pros and cons to that.
Yeah, I'm asking because I had experience with propagating rare species of Astragalus, and I provided ideal condition for them. They thrived, they flowered like crazy, but they didn't produce any pods because I didn't have bumblebees that pollinate them.
Yeah, so we really haven't done a whole lot of studies on the pollinators. I only know Melissa blue butterfly because you can see them around all over the place. I have seen, I know they're not native, but honeybees that were pollinating them. And then I have seen some flies that I just don't know my insects all that well.
Yeah, so you should add this as an important factor to
Will do. Thank you.
Yeah, to success of this, because if say we provide ideal conditions for the plant, but the area is surrounded by cultivating fields, there will be no insects, no fruits. This is an important factor.
Yes, it is.
All right, thank you.
Thank you. There.
Well, as a continuation of that, I was wondering about companion plants, and if you've looked at what companion plants, for instance, that they would encourage more pollinators, and the other thing that went through my mind was now the dam is going down, will that make more wet areas further down the Klamath that can be used?
So a couple things. We did collect species neighborhood compositions. It was not part of my thesis, though I would love to do that analysis, but it is in the works. I don't have any results from that though. And also the upper Klamath Basin where Astragalus is, none of those dams are actually going down. So I'm not so sure if it can survive in the colder weather, more towards the coast. I just don't know. I did talk to a U.S. Fish and Wildlife Service hydrologist, and he believes that there is going to be at least some impact in the upper Klamath Basin from the removal of those dams, though. Yeah.
So it looked like that you're
It doesn't really make any difference. Oh, okay. So it looked like that the survival rate of your planted plants was 10%?
Over 10 years.
Over 10 years.
Over
Okay.
10 years.
And so that does support the whole idea of reinforcement.
It does. So first of all, those are the oldest known plants, I believe. I don't think anybody else has monitored plants for that long. So they are the oldest known plants. But other studies, when they, translocate rare plant species, the survival rates are typically not very good for them. And so this is encouraging that these plants have survived that long. According to 1 out of 1 study, yes.
What was
It's okay, yeah.
what was the survival rate of the extent population?
So those weren't as old, as you could see by the statistical models there, but I think they were up to age 6 and 36% of them survived. Yeah.
So I have 2 questions. I enjoyed this talk a lot. I'm not familiar with plants all that much, so these might be silly questions. The first one is, are the fungi that are found in the mycorrhizae, are they unique to specific species? And can you inoculate with a near relative from?
I'm not 100% on that. I don't know a whole lot about mycorrhizal species or anything else like that, but there is another student that is working with mycorrhizal fungi on Astragalus applegate. So maybe we could have him come talk one of these days. Jonas Satinko, he's an awesome guy. Yeah.
And my second question is about the seed bank. I know with some plants, there's areas where there's a disturbance, they like turn up some dirt, and a plant that was thought to be locally extinct pops up from that seed bank. Is there any idea of how long seeds remain dormant for applegate?
Yeah, so when I did the math on all of this, I had to estimate how long seeds survive in the wild. And I used 5 years, 10 years, 5 years, 10 years, and 25 years, I believe. And I found that both 10 and 25 years, the reproduction rate just became so incredibly small that it just didn't seem realistic. So I assumed that it was a 5-year survival rate of these seeds. But yeah, like the Humboldt milkvetch, thought that was extinct for, I think, 100 years, and then all of a sudden it just popped up. Yeah.
Thank you.
Excellent question. Can I follow up on the seed bank question?
Of course you can.
You suggested that you were counting in the chart we had to memorize. I memorized it, and it talked about seedlings. You measured seeds and contribution to the seed bank, and then whether they should. How is that all modeling, or did you actually measure count seed bank?
All modeling.
There was no investigation into counting and determining what was.
No, so there's the best guess that I could make. Yeah.
Well, that's really interesting. And then off that topic, did you say discovered or rediscovered in 1980? Is that the first
rediscovered?
So
yes. Yeah.
So, extinct. Something happened, brought it up above ground again, and they found it.
Exactly. Or maybe they just went out to a site that nobody really, no botanists really had ever been, and they were like, "Oh yeah, here it is." Yeah.
And then note, very similarly to this Astragalus, here at the, what do you call the airport? The Redwood. I don't know the name of the airport is.
Yeah, at Arcata.
But our airport is a rare plant, Sedalcia oregana. And the same type of situation exists where it's so plentiful and so rich out there because of the altered disturbance regime from mowing. It's like, wow, how do we predict and how do we maintain that? They hit it on the head and it's doing really well out there. How can we make sure it continues? And that's the same thing here. You have to describe the mowing and make sure they do it the same every year.
Yeah, I mean, so I went out and, they drive their trucks on top of all the plants and they're not botanists. They don't care as much as like somebody like we do. But yeah, I mean, just the fact that it's mowing, I wish that there was more water out there, but again, you can't have sitting water on the landing strip or else airplane's gonna skid out of control, right? So that is a limiting factor of airports. But we also found that, at least when I was out there, every single year except for 1 year were extreme drought conditions. So I might have some biases towards that water retention on site. I wish I would have seen it, a few years prior when there was a lot of water happening.
Thank you.
Mm-hmm.
How many years of observation were the models based off of?
So the IPMs were 5 years or 4 transitions. And so I built models, annual transition models. I also pooled all of the data together for a pooled transition. And I did this for all sites except for Miller. I didn't have enough annual samples to do any sort of relevant statistical modeling. So I had to pool all of that data together. And then I did an overall model for like the species wide. And so those were the ones that I did. So 5 years total years. We continued to monitor the species past 5 years, but we changed the monitoring protocols. So I couldn't use those additional years in the IPMs, but I could in the translocation reinforcement project. Yeah.
Hey, Sean, you have a couple questions on Zoom if you're ready for them.
Yeah, go ahead.
The first one here is from Jim. His question is, at what height are they mowing?
I don't know the specifics of that. Probably, yeah, I don't know. Probably 4 to 6 inches, I'm guessing.
Great, thank you. The next one is from Ada. The question is, do you think it is likely that there are undetected populations, in parentheses, there's lots of private land out there, and then can new populations be established nearby and the land being exposed by the removal of the Klamath dams?
Yeah, I mean, there's always a potential for new populations. Obviously, private owners are a little weary of saying, yes, I have Astragalus applegate on my property. They, don't really want government involved on their property. So there's always a possibility that there's more. And again, as far as the dams go, the dams in the area are not being removed. It's more towards the western side, the California side of things. And I don't know enough information about that project to really say much on that. If there's suitable habitat, similar habitat, then sure, we can definitely try to establish a new population there.
Thank you. And last question is from Ronald. And the question is, does this milkvetch depend on fire to germinate?
No, it does not. Fire is definitely a way to cut down on competition with other species, or potentially is. At Uwana, there was actually a fire there, and we were really hoping that it burned at least part of the habitat of Astragalus applegate, but it was too far to the west for that to happen. So we can't monitor that. All right, one more question in the
Historic. The historical spread or locations, the range, that's the word, of the vetch known for this milkvetch?
No, there was only one observation of it by Barnaby, who found a lot of different plants around the area. And there was, I think that's the only real written observance of it. And so they didn't do a full scale.
Was that in the Klamath area?
That was in the Klamath area. Yeah. And that was in the 1960s, I believe, 1930s, maybe. I forget the exact date of it.
So they fix nitrogen in the soil.
Correct. Yes. Yeah. Yeah. If they want to be more sustainable farmers, definitely.
So could you say a few words about how you feel like this analysis that you did for your thesis can be applied to other similar species too?
Yeah, so I mean, if you really want to dive into the math, now I use a rather complex model. There are simpler population models out there to use, but anybody that wants to collect long-term data in their own garden and kind of test out where they're planting these different species in their own garden can do this analysis. Yeah, I mean, anybody can do it. Just takes time, you know, you need sufficient amount of years, you need to be consistent about it and all that good stuff. Yeah.
Okay, looks like there's no more questions. Is that? Okay, well, thank you very, very much.
Thank you.
Thanks, Barbara. Thanks, everyone on Zoom.